Thank you all for joining us today for this webinar. We're going to discuss a new approach to rare skin disease drug discovery. I will let you know that we are recording this webinar today, and we will
join the meeting
afterwards. We do ask that everyone who's joining, please turn off your camera and your microphone, just to help with the bandwidth and to keep everything clear and easy to listen to during the presentations. We will be having questions and answer period at the very end of the webinar. If you have questions that come up during that time, you're welcome to put them in the chat, and we will bring them up later at the end. So as I mentioned, this is a new approach to rare skin disease drug discovery. As many of us know, it's very difficult to make rare skin diseases a high priority for medical professionals and researchers. Patient organizations that are looking to increase research and find treatments for rare skin diseases often face challenges of scale, scarce data, and political issues. Today, we're joined by Rarebase, which is an innovative biotech company that is revolutionizing drug discovery. They combine genomic science and innovative high throughput technology to accelerate the discovery of potential disease-modifying therapies. Rarebase has identified promising drug candidates from initial work in neurological diseases and are now poised to use the same approach to speed up discovery in rare skin diseases. Today, we have two presenters. First, we have Dr. Joyce Teng, who is a professor of dermatology and pediatrics at Stanford University with clinical and research focus on genetic skin diseases, followed by Christopher Moxham, PhD and CEO and Chief Scientific Officer at Rarebase. Thank you all again all for joining us. Please turn off your cameras and your microphones, and I welcome Dr. Joyce Teng.
Okay. Am I able to share my screen? Okay, now I should be able to. Wonderful. Okay, I'll put this back. Are you seeing the screen okay now?
Yes, perfect. Thank you, Dr.
Okay,
Joyce.
Wonderful. I'll go ahead and begin. Thank you very much again for inviting me to the webinar today. And I'm going to talk about my favorite subject, genetic skin disease and drug discovery. I'm a pediatric dermatologist at Stanford. And here my disclosure, I do work with several different disease organizations and receive funding support for my research in the area. So just to give you a brief overview about my clinical practice, the right lower corner of the slide is the original Lucille Packard Children's Hospital. And on the left side is the new building, just got added on a couple of years ago. Aside from taking care of children with generalized skin disorders, I do have several subspecialty clinic focusing on genetic skin disorders, especially epidermolysis bullosa vascular anomaly, and several specific diseases. And many of you are familiar with a national organization for rare disease in NORD. There are more than 7,000 rare genetic diseases are known, about 1,200 registered and affecting 30 million people. And although these are known as rare diseases, it uses a significant amount of healthcare resources. So discovery of therapy for these patients are very important. As a dermatologist, I'm going to focusing on those genetic conditions with the skin manifestations. They're known as genodermatoses. Over 10% of genetic disorders with skin manifestations, because there are so many, they're categorized into those that are associated with malignant potential, those that are affecting skin barrier function. They're known as keratinopathy, hair nail development, pigment changes, or they can cause skin fragility, loss of the mechanical tensile strength. And some of the genetic conditions have increased vascular presentation on the surface or increased photosensitivity, for instance. All of these genetic disorders the research especially supported by an institute known as NIAM within the NIH. And as you can see in the pie diagram, majority of these disorders do affect the skin barrier function as well. So this is give you a little glimpse of genetic disorders that we see in the clinical setting. In the dermatology field, we're very visual, rely on visual recognition. Pattern recognition is an important part of our specialty training. So, and for example, this young baby on the right upper hand corner with these pigment disorder at birth, and we recognize these kind of pattern, and usually as a trained dermatologist, we think about possible associated brain abnormality or endocrinopathy. So skin is not only the largest organ in human body, but it's also a window into our overall health as well. So this slide, and just to give you a little glimpse at some of the genetic disorders that impair skin function specifically. The left hand side here is a simple cross-section of the skin. It looks very simple with the basket wave stratum cornea on the surface and a superficial layer that's less than a millimeter thick and epidermis. But it is actually a very intricate structure with many complex protein and cells and organized in a very unique, specific pattern. And more than 40%, more than 40 genes in this area, the mutation can lead to human diseases and have significant impact on human health. And here's a couple of different examples. If there's a single amino acid mutation, for instance, to this transglutaminase 1 protein on the surface of the skin, it can lead to significant a disruption of the skin structure and lead to quite detrimental skin diseases, and sometimes it can lead to increased mortality as well. If we move down on the skin structure and look at the junction where the outlayer of the skin attached to the deeper layer of the dermis, and you can see again, this is a very, very complex structure with many important protein weaved together to build our skin mechanical to provide mechanical strength to the skin surface. And in a single, again, a monogenetic mutation that impair the function of this keratin 5 can lead to significant skin fragility and blistering and many of the downstream skin clinical manifestations. So genetic disease, as many of you, can be passed down from generation to generation. However, the hereditary pattern can be quite different. Sometimes when there's what we know as gonadal mosaicism, some of the kids are affected, but others don't. And when parents have a certain part of their differentiated mature tissue affected by some of the cells that carry genetic variant, these type of genetic disorders do not really pass down to the children. So genetics are complicated, and this part of my talk really highlights the importance of genetic testing and mapping. So I'm going to use one of the example of a somatic mosaicism to discuss about this point a little further. So I see a lot of patients with vascular anomaly. For instance, one of these vascular anomaly is known as venous malformation. And now we know that the majority of patients with venous malformation carry a somatic mutation known as TIE2 or tyrosine kinase. It's a receptor that sits on the surface of the cells. And with an increased number of venous malformation patients that we test, and gradually this phenotype, a genotype pattern start to merge. And now we know that if you a patient that carry a genetic mutation in the carboxy terminal of the protein, for instance, that's closer to the cell membrane, and it may have a certain type of a venous malformation. And if they have it, have a mutation that in the more distal end of the C terminal, it can lead to more extracutaneous disease manifestation affecting the GI tract and the other organ system as well. So this is known as a genotype phenotype mapping. This is extremely important in the genetic disorder clinic because we can, based on these genetic variants and how they present and predict how severe the disease is, and whether there's going to be presentations in extra cutaneous organ system. So rare disease patients are challenging sometimes that we don't know, and especially when the phenotypes, their variability to their phenotypes. So it is always helpful to work with a disease organization and to understand the patient's need and the variability of their clinical presentation, so we can provide as much supportive care as possible. Over the years, I have had the privilege of working with all of these rare disease organizations, and they're so important in terms of provide education, identify information center of excellence to provide the help and support that patients needed. And they're also really important partner for clinical trials and research. And so with the advancement of genetic testing and almost every week now, there are a couple of new genes discovered for certain unique genetic diseases. Based on this gene discovery, protein-based, cell-based, or gene-based therapy start to emerge just last month. And, we have now the very first gene therapy for epidermolysis bullosa, and we start to provide those type of therapy to our patients. So the field is advancement in these fields are really extraordinary and amazing. I dedicated most of my professional career and study this mTOR pathway, for example. This is a very important homeostatic pathway because it senses the nutrients, hormone stimulation, and cell proliferation signal, and it passes it down to inside of the cells and drive the cell to proliferate or stop the cell from proliferate. So it's a highly regulated system, almost like a car that was gas pedal and the brakes. And one of the important regulator within this pathway is the TSC tuberous sclerosis complex 1 and 2. I'm just going to show you a quick example of a poster child disease of those patients that carry mutation of the TSC 1 or 2. They're known as tuberous sclerosis complex. A single mutation within this complex can lead to very complex human diseases that affecting the kidney, the lung, the heart, and the CNS system. However, as a dermatologist, we know these diseases really well because young children at birth may present with these white spots on their skin, and with these white spots on the skin and 2 or 3 of these tiny little red spots known as the angiofibroma on the skin, we're able to make a very early diagnosis. So these are the major clinical diagnostic criteria. And these angiofibroma, although they're not life-threatening, that they can really be life-impacting, as they cause disfigurement and bleeding. And the standard treatment was surgical removal or ablative laser treatment. However, patients oftentimes require generalized sedation, and they recur very quickly. So we know that there's a compound known as rapamycin that used to prevent kidney rejection after organ transplant that has been on the US market since the early 80s. And I thought that since this is an inhibitor for the TSC complex, and there's a possibility maybe we can just compound these medication and use it for patients. So when medication in the US, at least, that we are able to access for any medical indication, there's a possibility to do these type of a pilot study. So this is 6 weeks after I applied a compounding cream on one of my patients' face, and these are all published. And this is a couple of weeks after, and you can see that the efficacy of the treatment really sustains. And now, we're very exciting. Last year, this therapy is finally received FDA approval, actually, for the treatment of facial angiofibroma. So this is one example when we know the target and we know how to drug the target. But what about some of the other heterogeneous disease that we see in clinic? There are over 40 to 50 genes that have being identified associated with vascular anomaly. It's such a heterogeneous group. It's very difficult to treat. So do we have another way to find perhaps a common target that we can use to treat these heterogeneous population? So another way to figure this out is that, of course, I'm simplified 10 years of research is to draw blood, and we look at the genes that in the blood. Chris is going to talk about these a little more as well. And so some are upregulated and some are downregulated. And then we do these complicated pathway analysis. Then after we figured out which pathway, for instance, is driving the disease process, and we can incorporate a drug discovery platform. And for instance, that for this particular disorder, we identified that mTOR pathway is probably a very important druggable pathway, and we can use sirolimus to treat these vascular anomaly. Now, for the past 7 to 8 years, sirolimus has emerged as the first-line treatment for this very, very heterogeneous group of patients. So in summary, hopefully I give you a little taste of how complicated the genetic disease can be, and however, that with the advancement of genetic testing and different computational platforms, it make this lengthy drug discovery pathway a lot faster. It used to take about 2 decades, moving from early drug discovery, target identification, through the preclinical phase and clinical development. We have to do phase 1, 2, 3 to validate the efficacy and safety of a drug. And we may start with thousands of compounds. And if each compound has to go through this pathway, it takes a long time. So there are possibly shortcuts. And one of the example is the drug repurposing we just discussed. So I'm going to end my talk here and pass on to Chris. Thank you very much for listening. I'm going to stop sharing.
Great. Thank you very much, Dr. Teng. That was great. So I'll pick up where Dr. Teng left off, and that is how can one take an approach to discover effective therapies, but perhaps even with a faster path to the clinic. And start a little bit talking about the drug discovery pathway, not in its entirety, as Dr. Teng just kind of showed you a schematic of the entire path from drug discovery through clinical development and FDA approval, but rather more of a thought process around how does one think about the therapeutic rationale? That is, are we interested in a symptomatic that purely treats the symptomatology of the rare skin disorder, or are we looking for a disease-modifying approach? And I guess one would argue that, if you can come up with disease-modifying approaches, these will be truly life-changing for patients and people living with a rare skin disorder. And so to do that, one typically focuses on the root cause. And Dr. Teng highlighted some of the work that she's done to identify the root cause of rare skin disorders, particular skin disorders, and how one might start to target those. So among the different modalities that, again, Dr. Teng mentioned, you have to think about sort of multiple different factors that go into each particular modality. And whether it's a gene therapy or an antisense oligonucleotide like an ASO, or small molecules that we have all probably seen as pills, and now with cell therapies, one has to think about the convenience of how is that administered, and the effectiveness of that. And so the gene therapy that's been approved today for EB is a local administration. And if the skin disorder is highly localized, then that may be an effective way to go. But if it's not, then you may want something that's more widely distributed. And so that brings you to the next sort of consideration, which is the relative biodistribution, or how well is a medicine distributed throughout the body, and particularly across the entire skin surface. And then there's the element of tunability. That is the ability to increase the effect or decrease the effect, particularly if you need to have more effect, is their ability to increase it, but also if there are safety signals that start to emerge, you would of course like the ability to turn that off, right? And so in the case of a small molecule, for example, you can stop taking the medicine, and that would reduce the amount of drug in your system and eventually turn down the effectiveness of the small molecule. And then lastly, as a drug hunter, if you will, the others relates to what's the relative experience of any one of these modalities in the drug discovery world, if you will, at large. That is there a relatively limited experience of using this type of modality in patients? And that begins to speak to more about the safety profile. That is, how well do we understand the safety implications of a gene therapy or an ASO in the long term, let alone the short term? And so as one considers all these different sort of parameters about thinking about what modality to go after, at least in my analysis, small molecules really offer an opportunity that have a very convenient administration. They are broadly distributed throughout the body, certainly have the ability to be distributed to the majority of the skin. You can tune the effect of a small molecule, as I said, by either increasing the dose or stop taking the medicine altogether to turn down the effect. And of course, there are decades of experience with small molecules being used as medicines, both in terms of their discovery, development, and then ultimately marketed drugs, relative to some of the other modalities that are listed here. And it's not to say that one shouldn't consider these other modalities, but rather in this type of grid and sort of considering all of these different things together, if one can achieve it, a small molecule, I think, will be a solution that a global population would likely benefit from most. So how does one measure effects on root cause genes? And there are a couple of different methodologies that one can do that. On the top is a method called quantitative PCR. And this simply allows one to look at a single gene at a time, or 3 or 4 genes at a time, to quantify how much of that gene is present in the absence or after treatment with a drug. There's a newer methodology that has been around for probably the last 10 years called RNA sequencing. And this allows one to look at the expression of many genes all at once. You can look at the expression of thousands of genes all at the same time. And so it gives you a bit more scale. On the topic of scale, in terms of what type of experiments does one do in the context of drug discovery? Again, you can either do single experiments with a single drug at a time, looking at a single gene using qPCR, or you can use a methodology that's called DrugSeq. And what this really allows for is that allows one to screen thousands of compounds at a time and look at thousands of genes all at the same time. And so it gives you a great opportunity to look for potential disease-modifying therapies across many diseases all at the same time. And in the context of rare disease, that ability to just try to discover therapies for many diseases all at the same time, I think, is really what our society needs. That is, and as Dr. Teng pointed out, there are many rare diseases, and 95% of patients living with a rare disease don't have an effective treatment. And so if we want to have a significant impact for the benefit of society in a shorter period of time, we can't go after these one disease at a time and go through that cycle of 10 to 20 years, rather find a way to parallel process, if you will, and pursue disease-modifying therapies for as many diseases in parallel as we can. So what could be that faster path to the clinic, right? And so my experience has been predominantly in the small molecule space. I kind of highlighted the various attributes of a small molecule therapy as it relates to a rare skin disorder, finding a therapy for a rare skin disorder. You can deploy this DrugSeq technology at scale, as I said, to interrogate multiple rare skin disorders all at the same time in a cell-based system. You can focus on root cause genes, but as Dr. Teng alluded to, you can also actually look at pathways or signatures of gene expression to infer what's been changed in the context of the disease, and then look for small molecules that return that gene signature back to a more normal phenotype. And of course, if one finds a small molecule that modulates that root cause gene and moves that expression of that root cause gene in the appropriate direction, one could potentially identify disease-modifying therapy. And again, they would have to move into clinical trial testing. So here's an example of some of this high-throughput screening in action, if you will. On the left-hand side are images of keratinocytes that are grown in a dish in a lab. And what you can see is that the cells form these very kind of flat cell-based structures. And the two colors that are shown here, one relates to a keratin 14, and Dr. Teng had highlighted this on that schematic of the skin that she showed. And the color in red relates to what's called a transcription factor that is unique to keratinocytes and helps promote their keratinocyte-like state. And so in the lab, what one can do is when you have cells that you believe are keratinocytes, you can stain for these different markers using antibodies that have dyes that produce these different colors to confirm, in fact, that as you can see here, virtually all of the cells have the green color, and virtually all of the cells have the red color. And that's what you would hope to have in your cell-based model that you're going to then screen compounds against. And so with modern day automation that exists within laboratories, one can screen a library of compounds, say 9,000 compounds or more, in pretty rapid succession, and identify, and using next generation sequencing to look at many genes all at once, using these sequencers that one can gain access to, and then understand when you analyze the data on the far right, you can begin to produce these plots. And these types of plots are, in one example shown here, are called volcano plots. And on a volcano plot, what you have are all of these dots are different molecules that were screened in these, say, in a keratinocyte. And in the first panel, we're looking at root cause gene number 1. And maybe we're looking for molecules that increase that expression. And so these would be molecules that are in this upper right quadrant of this volcano plot. And with modern day computational approaches today, one again can analyze very quickly the effect of many drugs on 20,000 genes all at the same time and produce all of these volcano plots very rapidly. And so today, the ability to not only test thousands of molecules in a screen, but now look at their effects across thousands of genes all at the same time, ultimately allows drug discovery scientists to find potential therapies for many rare skin disorders, for example, in parallel. And as I said, I think that's really what our society needs to really make a dent in the large unmet medical need that exists for people living with a rare disease. So just wanted to kind of orient you to how you can start to look at the data. And I realize there's a lot here, but I'll just sort of set the stage. So on the top, we have different compounds with different serial numbers, 1, 2, 3, 4 through 8. On the left-hand side, we have different root cause genes that have been identified by the medical and scientific community as the root cause genes that promote various rare skin disorders. And the coloring or the heat map that's produced within this grid then is indicates which direction a particular gene's expression was moved. Either with red, the expression was increased, and perhaps that's the thing you're looking for. If it's a more neutral color, more of that white color, there was no effect, or if there was a downregulation, it's more of this bluish green color. And what one can do, as you can see, quickly looking at the grid, is for any particular gene of interest, you may find compounds that say RB05 that really increase the expression of TYMS, and perhaps that's the direction you want to move that gene for a potential therapeutic. And as it relates to the types of compounds that can be screened, some of these can actually be what we call clinical candidates. That is, they can either be FDA-approved drugs, as Dr. Teng mentioned, but they can also be molecules that have gone into other types of clinical trials for some non-rare skin disorder, for example. They may have failed in those particular trials, but you can actually screen those molecules and see if they have any application in the ability to modulate root cause genes in the proper direction for a rare skin disorder. And the advantage of doing that is that with clinical candidates, these have already had some human experience. They've been dosed in humans. There's some knowledge of their safety and tolerability profile. And so again, this creates a fast path to get into a clinical trial for a rare skin disorder, for example. So as Dr. Teng finished up, I think it's just important to kind of put research results in the context of how that fits into the larger process of drug discovery. And so when you have your research results, and the research results many times will be looking at efficacy, that is, how effective is a particular small molecule in this case. And there are two sort of distinctions in terms of how molecules are characterized in terms of their pharmacology. And that is either through in vitro approaches, that is in cell-based systems, as I just described, and that's where you may have this heat map that points to changes in gene expression. You'll probably want to look at and understand whether that translates to a change in protein levels, because ultimately it's those proteins that Dr. Teng talked about in the skin that have to produce that functionality and provide that important barrier, if you will, and other functions. You can also ask questions as to whether there's a phenotypic rescue in a functional assay, and that may include obtaining patient-derived cells that harbor these genetic alterations. And then if you treated with your drug candidate, is there a particular rescue of the phenotype that those cells demonstrate? And that gets back to this important genotype-phenotype relationship, and then understanding whether a small molecule or potential therapy can actually restore that genotype-phenotype relationship back to a more normal phenotype. The other types of assays that one typically conducts in terms of looking at efficacy have to do with in vivo studies. That is, typically in animal models, one wants to demonstrate that you can, in fact, dose the animal with the drug. You can determine how much drug is actually in the blood of the animal, so how much exposure do you have? But then importantly, relate that drug exposure to the effect on, again, either the root cause gene, the protein levels, or some phenotypic characterization. But certainly the most important thing as we start to think about therapies is around safety. And safety is really the primary focus of the FDA as well. And so as one begins to submit regulatory documents and engage with the FDA, the biggest focus will be on what's the safety margin around this particular therapy. And if you were to move forward in terms of clinical testing, how can you ensure that you're going to monitor this, not only the safety, but also enter into those clinical trials in a way that doesn't put the subjects or the patients at undue harm in terms of how fast you start dosing at higher levels. So again, as Dr. Teng mentioned, there's a process, and importantly so, there's a process that, again, includes typically multiple clinical trials where one has the opportunity to demonstrate a robust and reproducible effect in terms of both safety and efficacy, such that when medicines are approved, there's a high level of confidence that there's been sufficient testing to not put the public at harm. So with that, I think as Dr. Teng and I both talked about, there is a very high unmet need within the rare disease landscape, but particularly in rare skin disorders. What I've shared with you are high throughput ways to begin looking for therapies for many diseases all at the same time. And that hopefully creates an opportunity for an unprecedented opportunity for patients and organizations to become more actively involved in promoting drug discovery in their particular areas of interest. And as we're all here today looking at and talking about finding therapies for rare skin disorders. So I'll thank you for your time, and Dr. Teng and I'd be happy to answer questions.
Thank you so much, both Dr. Teng and Christopher, for those presentations. We will ask for questions now. You can put in the chat, or you can raise your hand and I will call on you. It'll give you a moment. While we're waiting, I did have one question myself, Christopher. So you had mentioned that some of these molecules can come from not only FDA approved, but also those that are in clinical trials. So does that open it up to clinical trials that are happening globally and not just in North America?
Yes. So you can, again, kind of a savvy way to think about this is that, again, there are molecules that fail in clinical trials and other indications every year, right? And those molecules are kind of privileged in a way, in the sense that they already have all of the attributes that allowed them to go into human testing. And so if you can just reposition those for a rare skin disorder, for example, that creates a fast path to kind of take advantage of the fact that this molecule already has drug-like properties and find a way to get it into clinical testing, either locally in the US, if you will, or more globally.
Were there any questions from our guests today? Karen is asking that they have problems with reimbursement in the Netherlands. Do you think that this new idea will help us gather more evidence? Was this, maybe we'll ask Dr. Teng if you have a thought on that question. Do you see this in the chat?
Yeah, I think that, reimbursing and access to therapy is a global issue and a global problem affecting the rare disease community. And however, that I've seen a number of organizations work together, and it is really important to educate, in the U.S. regulatory board, including FDA, NIH, sometimes need to be educated about the impact of disease on patients' quality of life and many aspects of the disease and the suffering they experience. And we deal also with insurance frequently in the US as well. Once some of the therapy becomes available on the market, and there are many patients may still not having access to these therapeutic options. So we frequently do what's called the MT to MD discussion with the insurance company to educate them. So it is a process. I think this is when rare disease organization group becomes so important in terms of raising awareness and educate the public, scientists, and regulatory boards, and as well as insurance company as well.
Thank you. We have another question from Samuel Simmons. Are there any current or future opportunities to incentivize or help pharmaceutical and biotech companies gain interest in rare skin disease discovery? And how do, what role can and patient organizations play in getting focus on their rare skin diseases? Maybe we'll ask that of you, Christopher.
I think there are a variety of ways that patient organizations can influence either regulators or pharmaceutical companies in that question. I think, for example, the FDA has these patient-focused drug discovery days where patient organizations will come in and basically tell their story of how particular disease, a rare skin disorder, or a multitude of them, affect patients, their quality of life, affect their families. And those are really compelling stories. And I think are today having a positive impact on the regulatory landscape. That is the way in which the FDA looks at how we conduct trials and clinical testing for rare diseases, to continue to evolve it, to make it more appropriate in terms of how rare diseases affect patient lives. In terms of, pharmaceutical companies or drug discovery organizations, I think it's kind of the same thing. That is, the opportunity to meet with scientists from those organizations to help them understand the impact on quality of life, and also the opportunity, frankly, to bring benefit to patients. And as someone who's been in drug discovery for the last 27 years, my colleagues and I wake up every day wanting to find medicines for patients, and people like Dr. Teng want to treat patients every day. And so there's no shortage of individuals who want to help patients. I think it's really getting your message in front of them, helping them understand the unmet medical need and the opportunity to have an impact.
Thank you. Christina in India has some question. Christina, I don't know if you're comfortable asking this question directly. I don't want to misinterpret. I feel like you're saying you have some patients who are on trials, and are you worried about the safety of the trials that are happening right now? Or this is a question about how to spread awareness that trials are available?
Yes, thank you so much, Stephanie. Well, most of the, I think almost 95% of the patients in India are from the low income group, and all of them are uneducated. So just a handful of us where we are trying hard to spread awareness. But again, it's very sad over here because people don't understand our challenges. And I found that there are some people who are giving them some medicine because these people don't know what they are taking. All they say is we apply some cream and we are given some tablets. And suddenly they see all the scales are cleared. But again, after a few months now, most of them have developed severe pain in their bones and joints all over. And one lady says inside her stomach, I think the entire internal organs, she feels a kind of intense pain, and she cries for hours. In the night, she's not able to sleep. And now she's also losing her vision. So when I asked her, can you share as to what is the tablet you're taking? She doesn't know. She says it's white. So now that's becoming a challenge, and I don't know where and whom. And when she says, no, the hospital told me you are not supposed to share the details of the meds, what you're taking. Now this is becoming a concern for us. I don't know how to solve this issue.
Maybe I can give it a try. And it sounds like you're raising issues about how clinical trials are conducted. And this sounds like a patient who's on medication may have experienced adverse effects from the treatment, and perhaps that they're not properly addressed. And I don't know how clinical trials are set up in India, but we at a given time do at least 7 to 10 clinical trials simultaneously, both the systemic therapy as well as a topical therapy. So maybe I can speak a little bit from my perspective as an investigator and a clinician, as well as a scientist, and how we conduct a clinical trial. Usually in US, for instance, if we're starting with a clinical trial at our hospital, Lucille Packard Children's Hospital, and giving patient a therapy, we monitor the patient closely, and your patient is probably correct. Sometimes these therapy have to be conducted in a randomized fashion so that we're not biased. If I know, if I'm not blinded and these trials are not randomized, when I assess the patient about whether the drug is working, not working, the results could be biased as well. So this is why we conduct a blinded randomized study. And this is perhaps a reason that, your patient may not know what medication, what drug, whether it's a therapy or it's a placebo drug that he or she was taking. But regardless whether the patient is on active medication or not, any side effects, sometimes including a cold, a flu, a cough, runny nose, everything that happens during the clinical study should be properly addressed and documented. And especially the investigator, every clinical trial has a principal investigator. The principal investigator need to investigate all the adverse events, including the pain the patient are experiencing, and try to figure out whether this is a medication related, possibly related, or unrelated event, and therefore making the decision whether the treatment should be terminated, discontinued, and the patient should seek a care in a different fashion. So, I know that not all clinical trial participants are well versed in the about medical environment. However, they should be educated when they participate in clinical trial about their rights and what they should do. And they always, every participant of the clinical trial has the rights to discontinue and drop out if this is not the treatment for them, if they experience severe side effects.
Thank you so much, Doctor.
You're very welcome.
Thank you, Christina. We do have another question again from Samuel. Let's see. As a follow-up, with the American Academy of Dermatology or Society for Investigative Dermatology annual meetings be an appropriate forum for the patient leaders to engage with pharma and biotech companies to gauge interest? And are patients eligible to attend those meetings?
Yes.
Thank you, Dr. Teng.
Yes, you're allowed to participate all of these meetings. There are actually many others, like the Society of Pediatric Dermatology, for instance, has an annual meeting, and Pediatric Dermatology Society has a network called the PEDRA, Pediatric Dermatology Research Alliance, and meet every year. This year, actually in 3 weeks in Atlanta. All of these meetings are indeed, we strongly encourage all the patient support groups, and including patients participating, and there are also regional meetings, so don't overlook that. I know there are many different regional meetings, and oftentimes and they are offered as an educational session, and have a clinician speak to at those regional meetings, and have pharmaceutical companies participating about new drug discoveries as well. So there are many different venues, absolutely, for both the organization and patient directly.
Dr. Teng. Jennifer Austin, CEO of GlobalSkin, I see your hand up.
Thanks, Stephanie. Well, thanks first of all to our presenters. I think this has been really interesting. I feel like an hour is only just scratching the surface of what this all means. And it's a lot to take in. Me personally, I don't have a scientific background, although I am a patient advocate and I'm trying to digest everything that you've said. I've had the benefit of talking to my colleagues before this webinar, so I do know a little bit more about what Rarebase is about maybe than others that are attending. But you've given us a lot of information, I think, and a lot to think about. But if you were leaving us with a takeaway of what we can do to better support our patients and having access to innovative, novel therapies, or even just therapies that have been used in other ways. And I'll just say that my son has a port wine birthmark and has been treated with rapamycin. So I'm very familiar with the context of taking something that's been used in other therapeutic areas and using it in dermatology and seeing the impact of that. But what is the takeaway for us as patient leaders? What can we do to help our patients? What are the ways that we could work potentially with Rarebase or other researchers that are doing this kind of work to fast track potentially where there hasn't really been hope or there haven't been new treatments coming on the market? So I think that's what I really want to know, is just kind of leaving this, what can we do? What can I do?
Yeah, I'll maybe go first, Joyce.
Yep, go ahead.
Certainly you can learn more about Rarebase by going to our website, and we're happy to sort of talk about how we might engage. I think there's a real groundswell in terms of opportunity for patient organizations to promote and drive more attention in terms of drug discovery for rare skin disorders, for example. There are other nonprofit organizations that have been created, like Every Cure, that are actively looking at, for example, FDA approved drugs for repurposing opportunities. And so I think there are a variety. There are just several organizations out there now, I think, where voices are being heard in terms of the large unmet medical need. And as I tried to allude to a bit, technologies today can be applied in a way that you don't have to just think of this as one disease at a time. And that you can actually go after many of these at the same time to try to find disease-modifying therapy. So I would, encourage the organization to reach out to biotechs and pharma companies that work in this space already, and understand if there are ways to work together. But also look at nonprofit organizations like an Every Cure, for example, that have a really interesting approach to try to use FDA-approved drugs to reposition them for application into the rare disease space.
Chris, could you repeat that last? You said to look at a particular company or organization. Would you be able to put that in the chat, please?
Sure.
Thank you.
Yep.
Okay, maybe I can just speak from the clinical aspect and patient, because the interaction that I have had with, in the clinical care setting. So there are a lot that the disease support group can do in terms of finding a cure therapy for a particular rare disease. Before a therapy can be developed, there are many important steps. We have to define the population. We need to identify care gaps, and we need to learn how to stratify risks, how to prioritize. So, for instance, I mentioned many of these diseases have many clinical manifestations. What are the most, life-threatening sometimes, and what's the most important aspect of the disease, and how to engage patients. And the therapy is not going to come out in the market when we, cannot engage patient population to participate in clinical trials, and data gathering, understanding the natural history, and how do we communicate about treatment therapy once it comes on the market, and how to manage the care. So all of these are very, very important steps. And for instance, I can use an example for like a muscular dystrophy, for instance, or epidermolysis bullosa. And before FDA conducted these open sessions to have a direct dialogue with the patients, they didn't understand what is a really meaningful clinical outcome for this therapy. And so sometimes they focus on, you need to develop a treatment to heal the wound completely 100%. But when they start to have these open dialogue with patient group directly, and all of a sudden regulatory board start to become to understand what is clinically meaningful really is for patients. So all of these are really important, and we cannot achieve a successful therapeutic development without each one of these steps before bringing a drug to the market. there's really a lot the patient group can do.
Thank you. We're coming close to the end of the hour. I'm just going to ask this one final question from Anne-Marie in the Netherlands. She says that some trials for SLE show that the lupus in the skin improves, but there's no add-on research for SCLE and other lupus variants. But how can this patient organization help to address that issue? Do you have any thoughts on that, Dr. Teng?
Yeah, I think that sometimes as a scientist and we work on silo as well, and we might be focusing one area, and there are a lot of diseases that have been many different therapies in recent years. You might be aware have been developed for lupus, especially for systemic lupus, and many new therapy that have proven to be effective. And the skin manifestation or SCLE, and the main manifestation is on the skin itself. And sometimes there's a little bit of a disconnection, and in terms of using some of these systemic therapies, for instance, for skin disease only. But we're, with the new discovery of many these systemic therapy for skin disease only, like eczema, psoriasis, and people start to understand the impact of main cutaneous manifestation of some of the diseases. And a similar, a parallel disease I can talk about is epidermolysis bullosa, and then I mentioned in the early part of my talk, and there's a severe form of epidermolysis bullosa that shortens lifespan. It doesn't mean that patients with EB simplex, with a skin only manifestation, they're not suffering, they're not in pain. And a lot of the research started to gradually shifting to focusing on how to address this area of unmet medical need. So in a sense, I think SCLE is probably in a very, very much a similar model as well. And clinician scientists will just need to be more focused on the disease impact itself, and to think about what kind of a therapeutic algorithm and approach we should develop to address this area of unmet medical needs.
Thank you so much. I want to thank you both, Dr. Joyce Teng and Christopher Moxham from Rarebase, for this very interesting topic today. It really gives us a lot to think about, and I think really presents a very hopeful idea for some potential therapies and potential relief for some of the patients that our members work really hard for. So thank you very much. Just a reminder, this has been recorded and we will be sharing this recording. Thank you all.